Okay, so hello and welcome everyone to the last lecture, and today is the week 12 lecture and the practice session, the live session, and then on Sunday I will be taking the final practice class where we would be discussing mostly questions. So we would be doing that lastly on a Sunday, but as of now today is the last lecture class that I would be live session class that I would be taking and discussing with you the lectures that were taught to you in this week in brief details and solving a few question and answers for your assignments and the exam that is coming up very soon. Okay, so I am sharing my slide. So I guess my screen is visible, and okay, so let us begin. So basically what we have in today's class is about a few topics that were left behind in this course, or these are something you can call as the special topics in protein chemistry. So we have to discuss a few of the topics among this. So mostly I would be beginning with what was there is the oxidative stress on the proteins, followed by something related to enzyme action and protease degradations. Then some important points about IDPs, that is intrinsically disordered proteins, and finally we have quite a few things to note down about the viral proteins, and then we will discuss a few questions. So the moment we talk about oxidative stress, so that brings to our mind the first thing is that stress, and the next thing is oxidation. So how does oxidation take place? So one important member that causes this oxidation, or an important, you can tell, thing that causes oxidation is free radicals. So, so basically, oxidation is a very normal and necessary process that takes place everywhere in our body, in nature, anywhere, any things that we see. Sometimes it is referred to as the oxidation number that describes the degree of oxidation or the loss of electrons of an atom in a chemical compound. And these free radicals are your chemical species that contain one or more unpaired electrons in an outer orbit. They are basically unstable and highly reactive molecules, and they um, carry out your autocatalytic reactions. So majorly the examples of these free radicals includes your reactive oxygen species, ROS. That is a very important terminology. Under that you have your superoxides, you have hydrogen peroxides, you have hydroxyl radicals, then you have the nitric oxide, peroxynitride, and you have nitrates and nitrogen dioxide, etc. So how are these free radicals basically formed? So these are basically formed by your oxidation and reduction reactions during any normal metabolic process that is occurring inside the body, and the primary source is our body during the energy production. So the enzymatic metabolism of exogenous chemicals and drugs, that is another cause of formation of free radicals. It also free radicals are formed during inflammation. Then if your blood flow is low, then it can lead to your heart attacks, strokes. Then there can be oxidation can be due to your environmental contaminants, smoking. This is some formation of free radicals inside the body. Okay, then absorption of radiation energy that can lead to your DNA damage. So if you look from perspective that your body is made up of millions and millions of DNA. Okay, so it in that manner it actually affects it, and then it can be low levels of antioxidants, diets, improper diets, and all. Now, what is this reactive oxygen species? So, this ROS is a chemically reactive molecule containing your oxygen with a highly reactive under normal conditions. So, these are basically your bioradicals. So, if you see the example where it's showing that oxygen is taking a electron, and you get an oxygen radical, then you get an hydrogen peroxide and another electron with a proton, you get water and the hydroxyl radical, and followed by you get the molecule of water. Okay, so this reactive oxygen species can lead to arthritis, then stroke, heart attacks, cancer, aging, and all. Okay, so basically, the examples of a few reactive oxygen species includes your, as I mentioned, superoxide radical. You have the peroxidyl radical. You have the hydroxyl radical, alkoxyl radical. Okay, so these have different properties and symbols, and also these are under the category of the reactive oxygen species. Now coming to oxidative stress. Now what is this oxidative stress? That is basically the balance between or the imbalance that you tell between your antioxidants and your free radicals. So free radicals are basically mostly your reactive oxygen species. So under normal conditions, what happens? The cells are able to balance the production of your oxygens and antioxidants, and oxidative stress occurs when you see that there is a rising level of ROS or a decreasing level of antioxidants, and it is basically harmful because your this reactive oxygen species attacks your biological molecules such as your lipids, proteins, and the DNA involved that can lead to different lifestyle related diseases. And these oxidative stress have a very useful role in the physiologic adaptation and in the regulation of your intracellular signal transduction. So when functioning properly, free radicals can help fight pathogens to prevent different kinds of infections. So your oxidative damage of ROS is, it occurs on the membrane, that lipid peroxidation, damage to membranes and lipoproteins that causes oxidative stress, disease, and aging. And then it affects your DNA where the strand breaks, it causes mutations leading to cancer and other diseases. Then you have proteins where it leads to aggregation, fragmentation, sometimes cleavages, and your inhibition of different enzyme kinetics. And all of these are related to your aging, disease causes, and oxidative stress. So how does oxidative stress cause damage of proteins? So your oxidative attack on proteins basically leads to modifications in different sites, say your catalytic site or the active site are residues of your enzymes, and if not, it causes fragmentations and you say aggregation of cross-linked reaction products. It changes, it alters the electrical charge and the susceptibility to proteolysis, so mutations and others. So it causes a lot of different problems. Then you have sulfur containing amino acids that gets interchanged, and the thiol group specifically, they are very prone to your oxidation. So the oxidation of iron and sulfur by superoxide destroys your enzymatic function. Now, just a minute. Okay, so this oxidative stress causes different kinds of problems in the proteins, and often the oxidation of your iron sulfur by the superoxide destroys the enzymatic function of different proteins and all. And if there is a mutation in the catalytic side, the enzyme of the activity of the enzyme gets halted to an extent. Okay. Now, what are antioxidants? So, antioxidant is any substance that delays, prevents, or removes the oxidative damage to a target molecule. So, there is no universal best antioxidant. So, basically, their relative importance depends on which, how, and where the ROS is generated and what target of damage is measured. Thus, the job of the antioxidant is to neutralize the free radicals that can harm our cells or affect the DNA and all. So, for the antioxidative defense, we have the enzymes that catalytically remove your ROS. You have the superoxide dismutase, the catalase, the glutathione peroxidase. You have the normal peroxidases, hydrogen peroxidases. Then you have the proteins that remove your pro-oxidants, the metallines and the heme, the transferrin, ferritin, albumin, haptoglobulin, celloplasmin. And these are basically synthesized in vivo, like you have the uric acid, NADPH, coenzyme Q, bilirubin. Then you get some from the diet, vitamin diets A, C, E. So antioxidants therefore can decrease mutagenesis and thus carcinogenesis in two ways: by decreasing oxidative DNA damage and by decreasing the cell division. Now we move on to the proteolytic enzymes. Okay, sorry for the delay. Uh, so we were talking about the proteolytic enzymes. So these are basically enzymes that are involved in the degradation of your proteins, or you can call them as the proteases or the proteolytic enzymes. So these are industrially available proteolytic enzymes that are produced by the microorganisms, that usually mixtures of your endopeptidases and the exopeptidases. So from proteins via endopeptidase you get polypeptides, and from polypeptides via exopeptidases you get amino acids. So these are the list of the proteolytic enzymes. You have the papain, ficain, bromelain. You can read from the slide. Then you have others, like you have the chymotrypsin, then other proteases. Sometimes you use chemicals like cyanogen bromide, etc. Now your peptidases are of two types. One is endopeptidase and one is exopeptidase. So your endopeptidase digests internal peptide bonds, and your exopeptidase digests the terminal peptide bonds to release the amino acids, like you have the aminopeptidases and the carboxypeptidases. So these exopeptidases are very important to get an idea about the N-terminal amino acids and the C-terminal amino acids. So they cut at the terminal ends of your protein. Now these proteolytic enzymes illustrate the range of enzyme specificity. So enzymes are highly specific both in the reaction that they can catalyze and in their choice of reactants, which are called substrates. So an enzyme usually catalyzes a single chemical reaction or a set of closely related reactions. So in vivo, these enzymes catalyze proteolysis, the hydrolysis of a peptide bond. Now, in vivo, these what happens that trypsin cleaves on the carboxyl end of the positively charged lysine or arginine residues. Chymotrypsin cleaves on the carboxyl side of bulky aromatic and hydrophobic amino acid residues, and elastase cleaves on the carboxyl side of the residues with small uncharged side chains. Now the protease enzyme functions, so there are many functions that are required for the regulation of metabolic and cellular processes. So they are proteolytic, that help in digestion and catabolism of proteins. They catalyze the hydrolysis of peptide bonds and convert them to amino acids, which is then absorbed and utilized by cells. They are required for the blood coagulation, then in cell division, growth, apoptosis and migration, protein recycling and transport across membranes. They are involved in activation of precursor proteins and zymogens. Then they provide immune support and regulate the processes of tumor growth, metastasis and inflammation. And they help in wound healing and muscle soreness. Now, just moving on to intrinsically disordered proteins. So they lack a fixed order three-dimensional structure in the absence of macromolecular interactions, like when they're interacting with proteins or RNAs, and they form different coil-like structures or molten globule-like aggregates, and they have flexible linkers in multiple domain proteins, and sometimes considered as a separate class of proteins along with globular fibrous and the membrane proteins. So IDPs are basically very large important class of proteins, and they can be identified to participate in weak multivalent interactions in highly cooperative and dynamic processes, lending them importance in DNA regulation and in cell signaling. So if we speak from the structural aspect, so these proteins have many structures in vivo according to the cell's conditions, creating a structural and conformational ensemble. Therefore, their structures are strongly function related. So their structures are mostly function related. However, only few proteins are fully disordered in their native state. Now, disorder is mostly found in your IDPs. You also see these in well-structured proteins where they act as loops. So the term intrinsically disordered protein therefore includes a protein that contains disordered regions as well as fully disordered proteins. So their appearance matters to us mostly because the existence and kind of protein disorder is encoded by its amino acid sequence. So in general, if we speak that IDPs are characterized by a low content of bulky hydrophobic amino acids, and their proportion to polar and charged amino acids usually referred to as a low hydrophobicity. Now this property leads to good interactions with water. Furthermore, high net charges promote disorder because of different electrostatic repulsion and all, and we know that the disorder sequences cannot simply bury inside the hydrophobic core that fold into stable globular proteins. So in some cases, hydrophobic clusters you will observe that the hydrophobic clusters in the disordered sequences provide the clues for identifying the regions that undergo the coupling, folding, and binding that refer to different biological roles. And these are very difficult to even understand the structure based on the Ramachandran plot, because it was meant for your alpha helices, then the beta sheets, and the left-handed alpha helix and all. But this is a disordered region, so it becomes quite complex, and it has a very low content of your secondary structures. So based on the disorder annotation, we have the disorder databases. Then we can predict the IDPs by the sequences that cannot form the secondary structures and all. So there are different websites and databases that you can basically use to find out the portion that is the disorder. Then you have different, you can generate graphs. Okay, so from that, because these disordered regions are also difficult to be carried out, it's difficult to carry out X-ray and NMR spectroscopy on these particular structures. However, they have a few biological roles. So the first one is that these act as flexible linkers. So the region where you want to use a linker sequence such that they can freely rotate. They can freely rotate or move. So where they can freely rotate or move, you can use them over there. Also, these act as very linear motifs. So these linear motifs are basically short disordered segments of proteins that mediate your functional interactions with other proteins and biological molecules. So many roles in linear motifs are associated with cell regulations in control of cell shape, subcellular localization of individual proteins, and regulated protein turnover. So often these post-translational modifications such as phosphorylation tune the affinity of individual linear motifs for specific interactions. Now, relatively rapid evolution and a relatively small number of structural restraints for establishing novel interfaces that makes it particularly challenging to detect the linear motifs. But their widespread biological applications and the fact that many viruses mimic these linear motifs and hijack the host cell or the host machinery. Then you have the pre-structured motifs. So the first one was flexible linkers, that was okay. Now the linear motifs is fine. So pre-structured motifs are unlike your globular proteins. The IDPs do not have any specially um, disposed active pockets. So fascinatingly, 80% of the target unbound IDP is subjected to detailed structural characterization of NMR possesses linear motifs termed prismos that are transiently secondary structural elements primed for target recognition. In several cases, it has been demonstrated that the transient structures become fully or stable secondary structures. And the last part for today is about the viruses. So viruses are infectious particles. They have a genetic material and a outer coat. So it's a very small non-infectious obligate intracellular parasite. They are non-living, and these viruses are chemicals and by themselves cannot reproduce. So a susceptible and permissive cellular host is needed for viruses to reproduce. All viruses must make mRNA that must be translated to the host ribosomes such that they can produce their own proteins. So you have a DNA or a RNA, then you have the capsid protein, followed by you have a nucleocapsid. So this is a nucleocapsid that is engulfed inside the virus. Now this naked capsid virus plus your lipid membrane and then you have the glycoproteins gives rise to an enveloped virus, where you have the outer coat is the envelope. So that is also made up of a few proteins. If you look at the capsid symmetry, this can be either icosahedral or helical. And see, there is an attachment that is occurring over here, and this is based on tropism. It can be the lock and key model like you observe for the HIV virus. Then there is the penetration, then it undergoes disassembly where the entire material is out into the host genome, and then viral enzymes leads to the formation of the mRNA followed by the viral proteins. And this is a viral genome that is once again replicated and then it is sent out in the form of nucleocapsids, and then you release these viruses. So that is the entire mechanism of a virus. If you observe for the retrovirus, this is a virus particle and it has a single stranded RNA, two copies. Then it undergoes entrance and uncoating. Then the single stranded RNA is inside the uncoded core. By the reverse transcription, it leads to the double stranded DNA and it travels to the nucleus. Then integration into the host DNA, followed by it forms a provirus, and then transcription. So in this manner, your retroviruses function. So the outcome of cellular infection is that you have a virus, then attachment into the cell, penetration, then multiplication, then it can transform into a tumor cell from the normal cells. Then it can also lead to death of different cells by the release of the viruses via the lytic infection. There can be some lysogenic or the persistent infection as well, but these viruses are active only inside the host machinery, so the virus present but not causing harm to cell, it emerges later on as a lytic infection. So for immune response, you have the cell-mediated immunity that is important for recovery from viral illness, important for the protection from repeat exposure. So this is your number of virions. You have acute infection, then persistent infection, and then on it increases to a latent infection. So I think that much is enough from the theory part. Now we can talk about the question and answers. There is nothing much to basically discuss because all of these have been already taught to you in the lectures in detail. So anyways, I am just telling information that was not basically something apart from that. Okay. So let us talk about the question and answer. So you people, we have only one member. Anyways, you can answer the questions. Then we can discuss. Okay. So the first question is, which of the following oxidized forms of cysteine is most likely to be directly linked to protein damage? You can answer in the chat box. You say it's D. Sulfinic acid form, sulfone form, sulfonic acid form, and the sulfonic form. See, with the increasing levels of reactive species, cysteine is oxidized to sulfonic acid, but it can further be oxidized to sulfonic acid, sulfonic acid, and your right. So you have the sulfonic acid and the sulfonic acid. Okay, and this mechanism is basically irreversible. So the sulfonic acid form that is formed from the sulfonic acid is a irreversible reaction. So that because it's an irreversible reaction, it causes an damage to your protein. So that is why C is the correct sulfonic acid, not the sulfonic acid. Okay. Now dityrosine, nitrotyrosine are produced from tyrosine in the presence of various reactive oxygen species. The two biomarkers are generally detected by dash and dash. Okay. So how can you detect the two biomarkers? UV visible, fluorescence. Sorry, just a minute. Okay, you are saying A, UV visible and fluorescence. See, in fluorescence spectroscopy, you would be seeing that the intensity of dityrosine is enhanced respect to that of tyrosine, because in fluorescence you can observe that. So it can be detected by your fluorescence spectroscopy, and your nitrotyrosine you can detect using your visible spectroscopy. And nitrotyrosine shows two absorbance peaks at 278 nanometer and 350 nanometer. So that will be the correct option, C. Okay, let's move on to the next one. Which one of the reagents is not used for analyzing N-terminal peptide sequence? Cyanogen bromide, dancyl chloride, 1-fluoro, 2,4-dinitrogen benzene, and phenyl isothiocyanate. This one is pretty easy question. In Edman degradation, you use phenyl isothiocyanate. Okay, let us see what is your answer. The Sanger reagent can also be used, so it's 1-fluoro-2-dinitrobenzene. Dancyl chloride is also used, but cyanogen bromide is used for cleavage of the C terminal after methionine. Okay, next one. Okay, just a minute. I have an important call. Just a minute. Okay, anyways, you can try the next one. I'm just attending the call and coming in just 2 minutes. Yeah, you can just answer it and. Yeah, I am back. Okay. So what would be the answer for that? No answer. Let's see. What is it telling? The nuclear capsid of the Ebola virus contains DNA, as you know. Find the correct answer. Okay. Okay, no, this is not correct. So one is not correct. One is not correct. So A and B is not correct. The lethal virus is made up of 7 proteins encoded by its genome. Yes, that is correct. Matrix contents driving the virus. Yes, this is correct. I think that's ICO cylindrical nucleocapsid. So it's not circular in nature. This is incorrect. Yes, so C is the correct answer. Yes, that is correct. Yeah, good. The next one. Okay. Identify A and B. Oh, this one is easy. Identify A and B. This is an adenovirus. I'm just giving a hint. This is an adenovirus. Okay. We're telling C. So this is the first one is 5 protein and the hexane protein. Okay. No. B is right. It's an hexane protein, but A is a fiber. It's not a spike protein. Because it's a long fiber with a knob formation at its end that is extended from each of its vertex. The psychohedral capsid is made up of a hexane protein and a long fiber. Okay, D is correct. Which of the following processes do not occur in HIV? Okay. Now tell me, which of the following processes do not occur in HIV? What happens in HIV? Reverse synthesis of DNA from RNA is performed by reverse transcriptase. So reverse transcription occurs. So 2 means A and D is not correct. Okay. Now translation is the synthesis of proteins from your RNA. Since RNA carries your instructions for making new viral proteins, each section of the mRNA relates to making different parts of the HIV. So translation also occurs, but transcription does not occur. So 1, 2 and 4 is occurring. So 1 and 3 does not occur. What is 1 and 3? So 1 does not occur. Sorry, 1 and 4 does not occur. 2 and 3 is occurring. So 1 and 4, right? So C. The next one, an unknown protease cleaves the peptide sequence RA, G, K, F, P, Q and into 3 polypeptides. So name the specific protease. Okay, this is a good one. So which protease cleaves that? So where is it cleaving? After F, Y and after F and Y. Okay. So which protease cleaves after F and Y? Elastase, trypsin, chymotrypsin, pepsin. I mentioned this in my lecture. So which protease cleaves after your aromatic amino acids. Yes, very good. Chymotrypsin, very good. Okay, let's go to the next one. So this polypeptide is cleaved by thrombin and trypsin. So what is the digested fragments that you will get? So what is the answer? Thrombin cleaves after what and trypsin cleaves after what? Tricky question. And the next one is also the tryptidism of the peptide. GLK, PMR and WA. Yes, that would have been correct. But if you see that lysine is followed by a proline. So when an amino acid is followed by a proline, the enzyme cannot act on that because it is blocking the site of that enzyme for attack. So therefore, it will cleave over only here. Actually, thrombin cleaves after arginine, trypsin cleaves after arginine and lysine, but not when followed by a proline. So keep that in mind. So it is going to be another C. Okay, try solving the next one. A digest of an octapeptide contains this and this, two tripeptides and one dipeptide. So both the tripeptides do not contain arginine. So the dipeptide contains your arginine. That is clear. Okay. Now what is it? Where is the dipeptide? Tryptic digest. Okay, tryptic digest. So trypsin does not cleave arginine and lysine followed by proline. So keep that in mind and try solving it. It's a very easy one. See, it says 2 lysine, 2 methionine, 1 serine, 1 proline, and 1 arginine. So you have 3 cut sites. But if there are 3 cut sites, you should be getting 4 peptides or 4 fragments, but you are getting 3. So one must be followed by a proline. Let's see where do we have a proline followed by a means following the lysine or arginine. So we do not have over here. The proline should come after this. So we have over here and here. Now if it cleaves over here, you get here, here. So 1, 2 and 3, that is correct. If it cuts over here, so you get 1, 2. But there is no arginine in the tripeptide. So it is going to be B. See, KPK, MSK and G and R. Okay, the next one. So which amino acids are preferred in intrinsically disordered proteins? You have, which will you prefer in intrinsically disordered proteins? So C, phenylalanine, cysteine and arginine. See, if you are having a proline, that is going to disrupt your secondary structure. You know that. So you need to have a proline. Now, if you have a polar amino acid, but it is packed inside the core, so it forms stable non-polar. Okay, it's non-polar core structure. So the stable non-polar core structure gets prevented. So you need to have a proline. Okay, so you need to have a proline, and so in the presence of your polar amino acids, your stable non-polar core structure will not form. That is why glutamic acid and arginine is not favored. Sorry, it is preferred over in intrinsically disordered proteins in the formation
So
of
what about cysteine? Cysteine के लिए नहीं होगा include?
cysteine and glycine. I was just thinking that only. Glycine and cysteine. I feel proline, glycine, cysteine can also be correct. So these two I believe is correct. Okay. So I believe that is also a correct option. Yes, that is also correct. But phenylalanine, cysteine, arginine, no, that is not okay. And alanine, glycine, and phenylalanine, no, that is also not okay. Maybe it's not forming a helix part. Okay, so A and B both can be correct in this case. Okay. Because your proline and glycine mostly remains in the flexible regions only, so that is also another important fact. Okay. Okay, so and B can be correct. Okay, which among the following is not true about intrinsically disordered proteins? IDPs are proteins which lacks a stable 3D structure. IDPs are disordered only in localized regions. IDPs carry low densities of charged amino acids, and IDPs exist as free flexible conformations. See? Yes, so IDPs carry high densities of polar charged amino acid residues. So not low densities, that is right. Okay, the next one. Tau protein is polymerized into paired helical filaments and straight filaments, forming neurofibrillary tangles when the protein is highly what? Glycosylated, phosphorylated, acetylated, hydroxylated. So this protein is mostly found in the brain and 3 to 4 fold more what? Yes, so when they are highly phosphorylated, they form your tangles and helical filaments and forming your neurofibrillary tangles. So this protein is mostly found in the brain and is 3 to 4 times more hyperphosphorylated than the normal tau proteins. So yes. Now, which group of amino acid residues are all prone to carbonyl derivatives due to oxidative stress? Oh, tyrosine, methionine, cysteine. So, due to your presence of nucleophilic side chains, your amino acids like lysine, then histidine, threonine, cysteine, arginine are prone to form carbonyl derivatives by covalent addition to an aldehyde group, carbonyl group to the peptide chain. So, the lysine, arginine, cysteine, this is correct. 3. Oh, 3 is correct. And what else is there? Cysteine, histidine, and 3 and 4. So 3 and 4 is correct. So which one is incorrect for the fly casting mechanism of IDPs? So if you know about the fly casting mechanism, you can understand that over here you basically search for the site where your protein or the macromolecule or small molecule is going to bind, and then found that whether it is going to form a folded structure or not, because sometimes after binding they form a folded structure, so you check that. So um, search for and folding. Okay, so over here it also reaches a high affinity conformation and easy sampling for many conformation. Okay, so non-specific is not correct. Okay, that is what I am saying. So specific binding occurs, right? And now the last question is, which of the following compounds cannot work as antioxidant? So the first one is your glutathione that acts as an antioxidant. What is number 4? It's ascorbic acid. Number 3 is a kind of polyphenols, like you have EGC, ECG, EGCG, but that acts as an antioxidant. So number 2 is only the incorrect option. So that is all from the questions. And there is going to be an extra session where we will be just having a quiz session type on Sunday from 11 a.m. to say, let's say one and a half hours, 2 hours, and we will be discussing totally about the 12 lectures, mostly quiz, if and then there would be a few discussion sessions as well if you people have questions. You can do that on a Sunday, and then you can get ready for the exams and all. Okay, then. I think that's all for today. We can end the class then.
Okay, sir.